Unit 13, which integrates two major domains of biotechnology, plant tissue culture and genetic engineering. Plant tissue culture focuses on the in vitro cultivation of plant cells, tissues, or organs under aseptic and controlled environmental conditions. Genetic engineering, on the other hand, deals with the direct manipulation of genetic material to introduce desirable traits. Together, these fields form the backbone of modern plant biotechnology and are extensively applied in agriculture, horticulture, and pharmaceutical industries. For GAT-B preparation, this unit is highly important because it combines both conceptual biology and applied biotechnology. Questions are often framed around mechanisms like totipotency or gene transfer, as well as applications such as transgenic crops. Understanding the integration of tissue culture techniques with molecular tools is essential for solving analytical and application-based questions in the exam. This slide emphasizes a structured preparation strategy for GAT-B 2026, highlighting the importance of concept-based learning and consistent practice. It underlines the need to focus on MCQs and previous year questions, which are crucial for understanding exam patterns and identifying high-weightage topics. The slide also stresses a guided approach covering the entire processes from registration to counseling, ensuring that students remain aligned with the exam timeline. Additionally, it highlights the resources required for effective preparation such as easy to understand notes, mock tests, and doubt solving mechanisms. Regular updates and exam tips are essential to stay competitive. For an exam perspective, the slide reinforces that biotechnology preparation is not just theoretical, but requires continuous testing, revision, and strategic planning. Plant tissue culture is defined as the in vitro aseptic cultivation of plant cells, tissues, or organs under controlled environmental conditions. The fundamental principle behind this technique is totipotency, which refers to the inherent ability of a single parent cell to regenerate into a complete plant. This property distinguishes plant cells from most animal cells and forms the theoretical base for regeneration techniques used in biotechnology. The success of tissue culture depends on several factors, including nutritional composition and plant growth regulators, macronutrients NPK, micronutrients FEMN zinc, vitamins, and a carbon source like sucrose are essential for growth. Hormonal balance, particularly between auxins and cytokinins, determines whether cells undergo root formation, shoot formation, or callus development. Various culture types, such as callus culture, suspension culture, and organ culture, have diverse applications in clonal propagation and secondary metabolic protection. Micropropagation refers to the rapid clonal multiplication of plants using tissue culture techniques, enabling large-scale production of genetically identical plants. As outlined on this slide, it involves multiple stages starting from explant selection and sterilization, followed by establishment in vitro, shoot multiplication, rooting, and final acclimatization. Each stage requires precise control of nutrients and hormones, particularly cytokinins for shoot purification and auxins for root induction. This technique offers several advantages, including the production of disease-free, uniform plants throughout the year, independent of seasonal constraints. However, it also has limitations such as high cost, risk of contamination, and the possibility of somaclonal variation. Micropropagation is widely used in crops like banana, sugarcane, and potato, making it a key application area in commercial plant biotechnology. Meristem culture utilizes the apical meristem region zero point 0.1 to 0.5 mm, which is typically free from viral infections due to absence of vascular connections and high metabolic activity. These properties prevent virus replication, allowing the regeneration of virus-free plants. This technique is often combined with thermotherapy or chemotherapy to enhance virus elimination efficiency. The primary application of Meristem culture is the production of virus-free clones in economically important crops like potato and citrus. To confirm the absence of viruses, indexing techniques such as ELISA and PCR are employed. This method is particularly significant in agriculture because viral infections can severely reduce crop yield and quality, making disease-free planting material essential. Anther and microspore culture techniques are used to produce haploid plants, which can be converted into doubled haploids through chromosomal doubling. The source material is pollen grains, microspores, which are cultured to develop into embryos and eventually whole plants. This method significantly accelerates plant breeding programs by achieving complete homozygosity in a single generation. There are two main approaches: anther culture, where the entire anther is cultured, and isolated microspore culture, which involves culturing individual pollen grains. These techniques are particularly useful in crops like rice, wheat, and tobacco. Their application in breeding allows rapid development of pure lines, making them invaluable tools in modern agriculture. Embryo culture, also known as embryo rescue, involving culturing immature embryos to overcome seed dormancy or abortion, especially in wide hybridization process. The technique is crucial when normal seed development fails due to genetic incompatibility. It allows the successful development of hybrids that would otherwise not survive under natural conditions. Ovary culture, on the other hand, is used for the development of haploid plants and parthenocarpic fruits. Both techniques help overcome post-fertilization barriers and expand the possibilities of plant breeding. A classic example is the wheat cross maize, where embryo rescue is essential for hybrid survival and development. A protoplast is defined as a plant cell devoid of its cell wall. Protoplasts are typically isolated using enzymatic methods involving cellulase and pectinase, which digest the cell wall. Mechanically methods exist but are rarely used due to low efficiency and cell damage. Common sources include leaf mesophyll cells and callus tissues. Protoplasts are highly significant in biotechnology because they allow direct genetic manipulation and somatic hybridization. Since the cell wall is absent, foreign DNA can be easily introduced. Viability of isolated protoplast is asserted using staining techniques such as fluorescein diacetate (FDA), ensuring that only living cells are used for further experimentation. Protoplast fusion involves the merging of two protoplasts using agents like polyethylene glycol (PEG) or electrical pulses (electrofusion). This results in the formation of somatic hybrids or hybrids. Somatic hybrids contain nuclear genomes from both parents, while hybrids have the nuclear genome of one parent but a mixture of cytoplasmic components. This technique is particularly useful for overcoming sexual incompatibility between species, enabling the transfer of desirable traits such as cytoplasmic male sterility (CMS). A well-known example is the potato cross tomato hybrid Pomato. Protoplast fusion thus expands the genetic variability available for plant improvement. Somaclonal variation refers to the genetic variability observed in plants regenerated through tissue culture. This variation arises due to chromosomal rearrangements, mutations, and epigenetic changes during in vitro culture. While it may seem undesirable, it can be a valuable source of novel traits. From an application standpoint, somaclonal variation can be exploited to develop disease resistance or stress tolerant plant varieties. However, it also possesses a challenge in micropropagation, where genetic uniformity is required. Therefore, careful screening using field evaluation and molecular markers is essential to identify beneficial variants while eliminating undesirable ones. Synthetic seeds are artificially encapsulated somatic embryos enclosed within a protective matrix, typically sodium alginate. These beads mimic true seeds by providing physical protection and a microenvironment containing nutrients and growth regulators. The encapsulation process allows somatic embryos, which are otherwise fragile, to be handled, stored, and transported efficiently. The major advantage of synthetic seeds lies in their ability to enable clonal propagation, germplasm storage, and easy distribution of alike genotypes. They are particularly useful in forestry and horticulture, where uniform planting material is required. However, a key limitation is the low conventional rate into viable plantlets, which remains a major area of research for improving efficiency and commercial viability. In vitro germplasm conservation involves maintaining plant genetic resources under controlled laboratory conditions for both short-term and long-term storage. Short-term conservation is achieved by slow growth techniques such as reducing temperature or modifying osmotic conditions to limit metabolic activity. This allows cultures to be preserved without frequent subculturing. For long-term preservation, cryopreservation in liquid nitrogen -196 degrees Celsius is employed, where cellular metabolism is completely halted. Cryoprotectants like DMSO and glycerol prevent ice crystal formation that can damage cells. Techniques such as vitrification and encapsulation dehydration are used to ensure survival upon thawing. This approach is critical for conserving rare, endangered, and economically important plant species. Organelle DNA refers to the genetic material present in mitochondria (mtDNA) and chloroplast (cpDNA), which function as semi-autonomous systems within plant cells. These genomes are circular, double-stranded, and present in multiple copies per organelle. Chloroplast DNA typically ranges from 120 to 160 kb and encodes genes essential for photosynthesis, while mitochondrial DNA is larger and involved in respiratory functions. A key feature of organelle DNA is its maternal inheritance in most plants, making it useful for studying evolutionary relationship and phylogeny. Additionally, organelles' genomes plays a crucial role in traits like cytoplasmic male sterility (CMS), which is widely used in hybrid seed production. Advances in transplantiomics further allows targeted genetic modification of chloroplast genomes for enhanced expression and containment of transgenes. Satellite DNA consists of highly repetitive, randomly arranged sequences typically found in heterochromatic regions such as centromeres. These sequences include mini-satellites (VNTRs) and microsatellites (SSRs), which vary in repeat length and distribution across the genome. Due to their high polymorphism, they can widely used as molecular markers. Functionally repetitive DNA plays a role in chromosome structure, genome organization, and evolutionary processes. Although once considered junk DNA, these sequences are known to influence gene regulation and chromosomal stability. Their application in DNA fingerprinting and population genetics makes them essential tool for plant breeding biodiversity studies, and forensic analysis. DNA repair mechanisms are essential for maintaining genomic integrity and preventing mutations. Cells are consistently exposed to DNA damaging agents such as UV radiation, chemicals, and replication errors. To counterattack this, multiple repair systems have evolved, including direct repair, base excision repair, nucleotide excision repair, mismatch repair, and double-strand break repair via homologous recombination or non-homologous end joining. These mechanisms involve key enzymes such as DNA ligases, endonucleases, and polymerases, which collectively identify and correct damaged DNA. Efficient DNA repair is crucial for stress tolerance, survival, and proper cellular function. In plants, enhanced DNA repair capacity is often associated with improved resistance to environmental stresses. Gene expression is regulated at multiple levels, including transcriptional, post-transcriptional, translational, and post-translational stages. Transcriptional regulation involves promoters, enhancers, and transcription factors, while post-transcriptional control includes RNA splicing and stability. Translational and post-translational modifications further fine-tune protein protection and activity. In addition, epigenetic mechanisms such as DNA methylation and histone modification play a crucial role in controlling gene expression without altering the DNA sequence. RNA interference is another important regulatory mechanism that leads to gene slicing. Together, these processes ensure precise control of gene activity in response to developmental and environmental cues. Recombination DNA technology involves the artificial combination of DNA from different sources to create new genetic constructs. This process relies on key tools such as restriction enzymes, DNA ligase, and cloning vectors. The basic steps include isolation of DNA, digestion with restriction enzymes, ligation into vectors, transformation into host cells, and selection of successful recombinants. This technology has revolutionized biotechnology by enabling the production of genetically modified GM crops, vaccines, and therapeutic proteins. It forms the foundation for modern genetic engineering and is widely applied in agriculture, medicine, and industrial biotechnology. Cloning vectors are DNA molecules used to carry foreign genes into host cells, facilitating the replication and the expression. Common types include plasmids, example PBR322, bacteriophages, cosmids, BACs, and YACs. Each vector type has specific advantages depending on the size of DNA to be cloned and the host system used. Essential features of vectors include an origin of replication (ORI), selectable markers, and multiple cloning sites (MCS). Expression vectors also contain promoters to drive gene expression. The choice of vector is crucial for successful cloning and depends on factors such as insert size, host compatibility, and desired application. Restriction enzymes or restriction endonucleases are molecular scissors that cut DNA at specific recognition sequences. They are classified into type 1, 2, and 3, with type 2 being the most widely used in genetic engineering due to their precise cleavage patterns. For example, EcoRI produces sticky ends that facilitate efficient ligation. Gene cloning involves inserting a desired gene into a suitable vector and introducing it into a host cell for replication. Screening methods such as blue-white screening selection, and antibiotic-resistant markers are widely used to identify successful clones. This process is fundamentally to genetic engineering and enables large-scale production of specific DNA sequences. Gene transfer in plants can be achieved through direct and indirect methods. Direct methods include biolistics, gene gun, electroporation, and microinjection, which physically introduce DNA into plant cells. These methods are useful for a wide range of plant species, including monocots. The most widely used indirect method involves agrobacterium tumefaciens mediated transformation, where the Ti plasmid facilitates the transfer of tDNA into the plant genome. This method is highly efficient for dicot plants and being adapted for monocots. Gene transfer techniques are central to the development of transgenic plants with improved traits such as stress tolerance and enhanced productivity. Transgenic plants are genetically modified organisms developed by introducing specific genes to desirable traits such as biotic and abiotic stress tolerance. Biotic stress resistance involves protection against pests and pathogens, commonly achieved through Bt crops that express Cry genes derived from Bacillus thuringiensis. These genes produce insecticidal proteins that are highly specific and reduce the need for chemical pesticides. Abiotic stress tolerance includes resistance to drought, salinity, and extreme temperatures, often achieved by introducing genes responsible for osmoprotectants like proline and glycerin betaine. Transgenic plants may also exhibit traits such as herbicide resistance and enhanced nutritional value as seen in golden rice. Advanced tools like CRISPR-Cas and RNA interference are now widely used for precise gene editing and regulation. This slide covers emerging and interdisciplinary areas in plant biotechnology, including bioethics, terminator technology, nanotechnology, DNA fingerprinting, and gene silencing. Bioethics deals with concerns regarding the safety and environmental impact of genetically modified crops, ensuring responsible use of biotechnology. Terminator technology, also known as genetic use restriction technology, produces sterile seeds, raising both economic and ethical debates. Nanotechnology involves the use of nanocarriers for efficient gene or drug delivery, improving precise and reducing damage to plant tissues. DNA fingerprinting utilizes molecular markers like SSRs and VNTRs for identification and genetic analysis. Gene silencing techniques such as RNAi and antisense RNA regulate gene expression by inhibiting translation. Collectively, these approaches contribute to crop improvement, biodiversity conservation, and forensic applications. Subscribe BioBrains and stay updated with the latest GAT-B 2026 preparation tips.